Hello everyone. Welcome to the session of Anatomy of Flowering Plants Part 2. And this is going to be the NCERT section 6.2 lecture. Disclaimer. Learning objective. And today's learning objective to understand anatomical structures of the dicot and monocot stems. So today we are going to study about the anatomy of the dicot stem and monocot stems. We will see the different layers of the cells and the tissue that are present in the stems of both dicot and monocot. Next learning objective, identify the key tissue arrangement in the both stems types. So we will see the types of tissue arrangement in the monocot as well as the dicot stem. Next, to compare the internal structures of the dorsiventral and isobilateral, that is monocot leaves. So dorsiventral, that is called as leaves of dicot, and isobilateral means leaves of monocot plant. So we will see the internal structure of both the types of leaves, right? Next, to understand the vascular bundle arrangement and tissue organization, we will see what types of vascular bundles are arranged in the stem and leaves, and what is the different types of tissue organization in this. Recognize adaptation and the structural modification in the different plant types. So the plants are so many types, so they do modify their structures and they have some modifications, right, to bear up with the different types of environmental changes through adaptation. So that also we were going to study about the anatomical adaptations. Index part. So there will be the anatomy of dicotyledonous and monocotyledonous plant, that is NCERT 6.2 section. And in that there will be anatomy of dicotyledonous stem 6.2.3, because we have already covered up the dicotyledonous root and monocotyledonous root in our previous lecture, right? So there will be anatomy of monocotyledonous stem, that will be 6.2.4 section of NCERT. Anatomy of dorsiventral dicotyledonous leaf, that will be the 6.2.5 section of NCERT. And the last, anatomy of isobilateral monocotyledonous leaf, that will be the 6.2.6 section of NCERT. Then we will summarize today's lecture in the summary section. Then there will be some NCERT based questions. And at the end, there will be some keynotes along with some interesting question that you are going to solve with your own. Okay, let us start today's session. Features of dicot stem. And this feature we are looking in one of the example of the dicot stem, that is sunflower stem. So to study the dicot stem here, what example has been taken? Sunflower stem. And TS means transverse section. You will cut the transverse section of the leaf with a very sharp blade, and then you will stain it under the some of the stain that is used to see the different anatomical structures of the plant, and then you see it under the microscope, right? So epidermis. The first thing when you will see the sunflower stem under the microscope, which layer you will see? Epidermis layer. So that is going to be the outermost protective layer of the stem, right? Stem, outermost protective layer is the epidermis. And how it is look like? Single layer of epidermis, and it is covered with a very thin layer of cuticle. Cuticle is a waxy coating substance that is present outer surface of the epidermis, right? So that is the cuticle. And stems do bear, different stems, they may bear trichomes and a few stomata. We will see what are the trichomes and what are the stomata. We have already talked about the stomata structure, right? So here you can see the epidermis. You can see the cells of the epidermis. This is the first layer of the cell that is called as epidermis. Barrel shaped cells, right? And it is having less amount of cytoplasm with a central placed vacuole and nucleus are placed aside of it, right? So this is called the epidermis layer. You can see that. And the outer layer of the epidermis, not the inner one, outer layer, external layer, that is covered with a waxy substance that is called as cuticle. So this is the first layer of the stem epidermis. Now, the next layer becomes the cortex, located between the epidermis and pericycle. So this is a very, very important point about the cortex, that from where to where the cortex layer is present. Just below the epidermis. Epidermis is single layered cell, right? The second layer of cells starts the cortex until pericycle. Okay? Between the pericycle and the epidermis is the cortex region. And it consists of 3 subzones, means cortex can be divided into 3 subzones. Okay. So we will talk about the trichomes also, right? But let us first discuss about the 3 subzones of the cortex region. Okay. The outer cortex layer is called as hypodermis. Outer cortex layer is called as hypodermis. It is the layer that is just present below the epidermis. Okay. Just below the epidermis is the hypodermis. And you can see composed of few layers of collenchymatous cells. Collenchymatous cell. What type of collenchymatous cells are, if you remember? The collenchymatous cells they do not have intercellular spaces. First point. If you see the parenchyma cells, then they are having the intercellular spaces, right? But collenchymatous cells, that is collenchyma cells actually, right? And if you see the structure of the collenchymatous cell, then their ends wall or the corner walls, there is a deposition of lignin substance. There is a deposition of lignin substance. And this lignin substance, what they do, they provide stretchability. They provide stretchability and also mechanical strength to the stem, right? And they are having a thick wall. Yes, their cell wall is comparatively thick than the parenchymatous cells, right? So you can see here the third point of this hypodermis, provide mechanical strength to the young stem. When the stem is young, it is herbaceous, it is green in color, but still they required some sort of mechanical strength. So this layer, collenchymatous cells, which is present in the hypodermis, or this hypodermis is made up of collenchymatous cells, provide mechanical strength to the young stem. Very, very important point. So it can be asked in the examination. This hypodermis is made up of collenchymatous cell or sclerenchymatous cell or parenchymatous cell. There can be multiple statement questions, right? And in one of the statement, this could be the statement regarding the hypodermis. So you have to remember this hypodermis is made up of which type of cells and what is its function. Its function is to provide mechanical strength, right? Okay, let's move forward. So here in the diagram, you can see the epidermis layer is there, and just below the epidermis, there are 3-4 layers of cell, red color, that is called as collenchyma. Collenchyma. And this collenchyma is made up of this hypodermis. Hypodermis. Okay. Again, we will talk about the cortex because the cortex has been divided into how many regions? 3 regions. So first region was hypodermis. Now the second region. The second region is the cortical layers. Cortical layers. And this is called middle cortex. Middle cortex. It is present just below the hypodermis, where the hypodermis and this cortical layer or the middle cortex is started. What type of cells are present here? Just see, consist of rounded, thin-walled parenchymatous cell. Parenchymatous is having very thin wall. Contain conspicuous intercellular spaces. Now see here. In the hypodermis region, there were no intercellular spaces because the cell was collenchyma. And collenchyma, the corners, of the collenchyma, they are having the deposition of the lignin. So there is no intercellular spaces. The cell wall is also thick. Just see the middle cortex region. This is having a different type of cell, right? Parenchyma. Parenchyma. And they contain the conspicuous intercellular spaces, means they do have intercellular spaces in between because they are having round shape parenchymatous cell. And when the cells are round shape, the wall of the adjacent cell will not build fit and there will some space left behind, right? So that is why they are having the intercellular spaces. Very important point. The third layer, the third cortical layer is the endodermis. Very important. Endodermis. Innermost layer of the cortex and cells rich in starch grain, also called the starch sheath. So this is single layer. Single layer. Endodermis is the single layer of the cortex and it is having rich deposition of the starch material. Starch material. And this is, as there is a lot of deposition of the starch in this endodermis layer, so that is why it is called as starch sheath. It is called as starch sheath. So here in the diagram, you can see the collenchyma, then comes the parenchyma, and then there is the endodermis layer. So you can see here all the 3 different types of cells that makes the tissue. Okay, filler tissue, or you can see the ground tissue. They consist of parenchyma, collenchyma, right? And the endodermis is again a type of parenchyma cells, innermost layer of the cortex, and they contain the starch grains. Next layer. So this is the 3 region of the cortex. 3 region of the cortex. Outer one is the epidermis, then comes the cortex region, and cortex is divided into 3 parts. The outer one is the, what we call it, outermost cortex layer, that is called as hypodermis. Hypodermis. And just below the hypodermis, there is a many layers. It is made up of many layers. Then it is called as middle cortex region. Middle cortex. And then the innermost is called as the endodermis. Endodermis. So please remember the 3 different layers of cortex region. Okay. Now we will talk about the pericycle. Present on the inner side of the endodermis. Just below the endodermis, there is a pericycle region. Pericycle. located above the phloem. And just below the peridermis, you can see here the phloem means the vascular bundle is present. The vascular bundle is present. So it is located above the phloem, this pericycle region. In between the phloem and endodermis, there is a pericycle and appears as a semi-lunar patches of the sclerenchyma. Now here you can see this pericycle region is sclerenchymatous. Sclerenchymatous, semi-lunar patches, right? So here you can see this is the patch. This is the patch of one layer is the endodermis, and just after the one layer, there is a patch of many layers of the sclerenchymatous cells that is called as pericycle. And just below this pericycle, what you see, the phloem is present. Means the vascular bundle start just below the pericycle region, right? And this sclerenchyma are what type of cells? They are dead cells. They are dead cells. They do not have any living cells in them, right? So this is going to be again another feature. Now we will see the vascular bundle arrangement. Now why this pericycle is very, very important in the case of roots, if you remember. What they give rise? Pericycle region in the case of roots. What they give rise? Because here it gave rise lateral roots. Lateral roots. Pericycle gave rise the lateral roots in the case of root, but not in the stem, right? Okay. Numerous vascular bundles are present in the case of dicot stem, arranged in a ring pattern. And what is the arrangement of these vascular bundle? Ring pattern. But are they present on the different radii or on the same radii? The question is this. If the vascular bundles are present in a circular pattern, so circular pattern could be of two types. Radially arranged or radial vascular bundle type or conjoint vascular bundle types. In the radials, the xylem and phloems are present on the different radii, whereas in the case of conjoint, the xylem and phloem are present on the same radii, right? So when we were discussing about the radial vascular bundle, they were present in which parts of the plant? Roots. Radial vascular bundles are present in the roots, but conjoint, they are present in the stem. Okay. So we are studying stem. So here will be the conjoint, means xylem and phloem will be present at the same radii, right? Let us see that. The ring arrangement is a typical dicotyledonous dicot stem. So this is the arrangement of the ring. That is one of the feature of the dicotyledonous stem. When you will study about the monocot stem, you won't see this radial arrangement. You won't see this radial arrangement of the vascular bundle in the monocot stem. So this is a feature of the dicot stem, that the vascular bundles are arranged in a ring form. So here you can see, this is the diagrammatic and this is the cellular figure. So here you can see this is the vascular bundle. This is again second vascular bundle, third, fourth, fifth, like this. So the vascular bundles are present in a ring form of structure. It is forming a ring, right? And in this cellular diagram, you can see one vascular bundle. This is the phloem region. This is the this cambium region. Then the xylem region. Now, if you see the pericycle, if the pericycle is continuous, and the layers are same, then it is called as homogeneous. And if the pericycle is, here you can see lunar shape. What is the layer of cells here? just single at the corner. Here one cell. Here is also one cell. But what is the center you can see? Many layers. So it is forming a lunar or the spindle shape like this. It tapers at the corner. And this, again, the pericycle will be like this, right? So see the pericycle. It is not the homogeneous, heterogeneous, right? Okay, let's move to the next. Characteristic of individual vascular bundle. Now we will see one of the vascular bundle of the dicot stem. Here, what type of vascular bundle? Conjoint means xylem and phloem are present on the same radii. They are located on the same radius, right? So that is a type of conjoint vascular bundle. In the case of radial vascular bundle, they are also present on the radius, but not on the same radius. Xylem will be present on one radius, phloem will be present on another radius, again xylem, again phloem. So there will be the alternating arrangement of xylem and phloem radially. Here also, the vascular bundles are arranged radially, but the xylem and phloem are present on the same radius. So that is a type of vascular bundle that is called as conjoint vascular bundle. A very specific feature, a very important feature of the dicot stem. Okay. Okay. Next, open type of vascular bundle. What is this? Open. Vascular bundle is open. It means between the xylem and phloem, there will be one layer of cell that is called as cambium. And you must have studied regarding the meristematic tissue. Apical meristem, intercalary meristem, and lateral meristem. Remember lateral meristem? So this cambium is actually the lateral meristem, right? So this cambium layer is a very, very important feature. As you all know, what is a feature of the meristematic tissue? Meristematic tissue divide continuously. They divide and redivide and form the new cells, new layers of cells. So similarly, this cambium layer, cambium divide outside as well as inside. Outside, a new phloem, and inside, a new xylem. So in this way, the xylem and phloem grow and totally they bring about the girth increase, right? So this cambium is very, very important regarding the growth of the stem. So if the cambium is present, then it is called as open type of vascular bundle. So conjoint and open, this is two features. Enlarged protoxylem. Protoxylem is located toward the center of the stem. There are two types of xylem, protoxylem and metaxylem. Okay. So here it is called as enlarged type of protoxylem. So what is the difference between the protoxylem and metaxylem? They are both the primary xylem, but they differ in their development timing, right? Protoxylem is the first form xylem. Remember, protoxylem is the first formed xylem, whereas the metaxylem formed later on. So which type of xylem is formed first? Protoxylem. And this protoxylem is located toward the center of the stem, means inside. And metaxylem towards the cambium, right? So if the protoxylem is toward the center of the stem, then what type of vascular bundle it is, or what type of xylem it is? Enlarged protoxylem. Enlarged protoxylem. And if the metaxylem is toward the center and protoxylem is toward the inner, towards the cambium, then it will be called exarch protoxylem. Okay. Okay. So this is the three important point regarding the vascular bundle of the dicot stem. Must remember, very, very important point. Conjoint vascular bundle, open type of vascular bundle, and enlarged protoxylem, right? These are the very important features. Every time the questions come from this regarding the stem, right? Feature of the dicot stem, medullary rays found between the vascular bundles. Between the vascular bundle, there are some arrangement of the cells, which is called as medullary rays. Consist of radially placed parenchymatous cells. They are having radial arrangement of the parenchymatous cells. So this medullary rays, what type of cells they are made up of? Parenchymatous cells. And pith, pith. This is the center region, occupies the center portion of the stem. Center portion of the stem. And this is composed of the rounded parenchymatous cell. What type of cell they are having? Parenchymatous. But what type? Round shaped parenchymatous cell. Contains large intercellular spaces. So in the center here you can see the pith. Here you can see the pith. Okay. So this is the structure of the dicotyledonous stem, and the example was the sunflower stem. Now we will see the different features of the monocot stem. Monocot stem. Here the hypodermis is sclerenchymatous, vascular bundle, conjoint and closed. The other layers are same, like epidermis, they are having the cuticle layer, that is the same, right? Hypodermis, here you can see there, what was the hypodermis made up of? Collenchymatous. And in the monocot you can see the hypodermis is made up of sclerenchymatous type of cells. Very, very important point. Sclerenchymatous type of cells. See the vascular bundle, conjoint and closed. In the case of dicot stem, they are having the open type of vascular bundle. Open type of vascular bundle. But here they are closed. Closed means they do not have cambium layer. In between the xylem and phloem, the cambium layer is absent. Vascular bundles are scattered in the ground tissue, each with a sclerenchymatous bundle sheath. So they are not radially arranged. Vascular bundles in the case of monocot, they are not radially arranged. They are scattered in the ground tissue, and each with the sclerenchymatous bundle sheath. Right? So see the arrangement. There is a epidermis, there is a hypodermis. Where is the cortex? Where is the cortex? Where is the endodermis? Where is the pericycle? Can you see these layers? No. These layers are not found in the case of monocot. There is no endodermis, there is no pericycle, and we are not having the cortex region. Okay, we are, instead of cortex, we are saying the ground tissue. And in this ground tissue only, all the vascular bundles are scattered. And see the type of vascular bundle, conjoint and closed type of vascular bundle, right? A prominent parenchymatous ground tissue surround these bundles. So what are these ground tissue made up of? This ground tissue is made up of this ground tissue is made up of parenchymatous. Now, why the cortex is not present in the monocot stem? Very good question, right? Why they are not having? If you compare the dicot and monocot, monocot are comparatively herbaceous plant, but the, the dicot are comparatively stronger, right? And they are more robust, and their height is large. They have to stand erect because they have to bear the branches, the leaves. See the monocot plant, grasses, they're not so strong, right? And they do not require uh, different types of the arrangement of the layer. Now, if you think about why they are having the endodermis layer, endodermis, why they are having this uh, pericycle region, why they are having the vascular bundle just inside it in the this region, but here they are scattered. This is a difference, right? This is because the dicot plant is having more complex structure if you compare with the monocot. They are herbaceous. They are herbaceous, but see the vascular bundle. They are more vascular bundle compared to the dicot, right? But the vascular bundle of dicot are more robust, more, it's, see the size, and they have to show the growth, secondary growth. Dicot have to show the secondary growth. So the arrangement of the vascular bundle should be in a ring type so that the growth should be unanimous all over the stem. How the growth occur if they are having the monocot is having the open type of vascular bundle? How will be the growth occur? Can you think about? Because these vascular bundles are scattered, they will not form any circular type of ring structure, right? We are discussing the why the cortex is not present in the monocot. Not only this, but why the all these systematic arrangement of the cell, systematic arrangement of the cortex, systematic arrangement of the vascular bundle, systematic arrangement of the endodermis. They divide in the dicot, they divide region specifically. But in the case of monocot, they are not a specific division. There is no endodermis layer, there is no pericycle layer. The vascular bundles are not in a circular form. They are scattered, right? And the vascular bundle is what type? Open type. They do not have to show any like secondary growth. So they are herbaceous plant. Okay. They do not show any secondary growth, but the dicot have to show a proper secondary growth. That is why the vascular bundles are circularly arranged, and they are arranged or they are placed in the inside the endodermis. They are having a specific region for the presence of vascular bundle inside the endodermis, right? And this endodermis protect the outer part and also the inner part. Pericycle and vascular bundles are protected by this, and they are having a specific things that when you will study about the photosynthesis, there is a trans anatomy of thing that will be bring by this endodermis layer, because they are having this starch sheet. Okay, so these are some of the region that the cortex regions are not present in the monocot. Some other features of the monocot: vascular bundle. Peripheral bundles are smaller than the centrally located one. Now see, the vascular bundles are not also of the same size. They are of the different size. Peripheral bundles are smaller. If you see the vascular bundle which are present at the periphery of the stem, outerward, they are smaller in size. Whereas the one that is present in the center, they are larger vascular bundle. Okay. But in the case of dicot, all the vascular bundle are of comparatively same size. Phloem parenchyma is absent. See, one more feature. Phloem parenchyma is absent. Water conducting cavities are present within the vascular bundles. Here you can see. This is the cuticle, then comes the epidermis, sclerenchyma, parenchyma. This is the ground tissue, right? And then comes the vascular bundle scattered within the ground tissue. One of the vascular bundle you can see here, right? They are having the sieve tubes, companion cells that makes the phloem. And what makes the xylem? Vessels, okay, spiral vessels, xylem parenchyma, and so these are making the xylem part, right? Vascular water containing cavities are present. You can see They are having large water containing cavities in the vascular bundle within the xylem part. This is schizolysigenous cavity. Okay. Now we will discuss about the dorsiventral leaf. Now before going to the dorsiventral leaf, there was like trichomes, where the trichomes were present. Trichomes are actually the modification of the epidermis. This is the modification of the epidermis. Thus, epidermis give rise a hair-like structure. Hairs-like structure. Some are unicellular, some are multicellular. And some trichomes are glandular also. They secrete some substances. So, glandular trichomes are also there. Now, what is the function of these trichomes? Trichomes are the hair-like structures. They do not allow the sunlight to fall directly on the surface of the plant. Okay. So, they hide the surface of the stem, or they are also present on the leaf sometimes. So, they hide the surface, and in that way, they minimize the transpiration. Right. And they also give sometimes a cooling effect to the plant. Okay. So these are some of the function of the trichomes, and they are present on the outer surface of the stem and leaves. Modification of epidermis layer. Okay. Coming to the leaf structure. Now we will study about the anatomy of the dorsiventral, means dicotyledonous leaf. Now epidermis cover both the surface of the leaf. Epidermis is present on the upper surface as well as the lower surface. Okay. Both the surface of the leaf. Now, adaxial epidermis. Adaxial means upper layer of the epidermis. Upper surface means this epidermis is present on the upper surface of the leaf. It has a conspicuous cuticle. The cuticle is more at the adaxial epidermis. If you compare with the abaxial, means lower epidermis, right? They may lack stomata. Stomata may not be present on the upper surface in the dorsiventral or the dicotyledonous leaf. Can you think about why the stomata are not present on the upper surface of the leaf in the dicot plant? They are mostly confined to the lower surface. See, dorsiventral means leaf. Yes, to prevent the water loss. Yes. Okay. If they will present on the dorsiventral leaf, the leaf which is exposed, upper surface is exposed to the sunlight. So there will be more transpiration. But the stomata are present on the lower surface in the dorsiventral leaf because the sunlight is not coming to the lower surface directly, right? So upper surface, they may lack stomata. So here you can see, this is the structure of the dorsiventral leaf. Epidermis is layer, you can see this, and the cuticle layer is above this. Now see the lower surface. Lower surface is having the stomata, right? This is called abaxial epidermis and this is called adaxial epidermis. The terms are very important. Sometimes in the examination, if they will not write the upper surface, they will write adaxial epidermis. Then if you will not understand what is the meaning of the adaxial epidermis, then you may not be able to attend the question. Or if you attend the question may be wrong even. So abaxial and adaxial. Please remember these terms. Okay. Now epidermis, abaxial, lower surface, bears more stomata on the adaxial surface, epidermis, and it is conspicuous cuticle, very less cuticle. Mesophyll in the dicotyledonous leaf, located between the upper and the lower epidermis, right? This layer, mesophyll layer, this is present between the upper and the lower epidermis. Now this mesophyll cell, what type of cells they are having? Parenchymatous cells. parenchymatous cells that contains the chloroplast for the photosynthesis. Okay. These parenchymatous cells are having a lot of chloroplast, and that are actually the cells that are involved in the process of photosynthesis. These mesophyll cells are doing the process of photosynthesis in the leaf. If you see the inner structure of the leaf. Two types of cells. Now, mesophyll cells are of two types. First one, palisade parenchyma. Palisade parenchyma. Adaxially placed means these cells are present on the upper side. Okay. The shape is palisade, means column shape. Made up of elongated cell or the column shaped cell arranged vertically and parallel to each other. Now here you can see these are the palisade parenchyma. Okay. Palisade mesophyll it is written or labeled it here. And see the lower surface. They are not the palisade. They are round shape and they are called as spongy mesophyll, having a lot of air cavities. But the upper palisade mesophyll, they are not having much air cavities. They are parallelly placed. So the chances for the air cavities are very less, right? Spongy parenchyma situated below the palisade parenchyma. They are confined most to the lower surface, situated below the palisade parenchyma. Consist of oval or round shape. If you see the shape, then they may be oval or round, loosely arranged cells. And due to the loosely arrangement of the cells, they are having intercellular spaces. They contain numerous large spaces and the air cavities in between the cells. And these air cavities are of very important because these air cavities are there for the exchange of gases during the photosynthesis and respiration process. Right? So here you can see the air cavities. These air cavities are connected with the stomata. Right? The air can come and comes to this. They can reach to this region. They can reach to this region. Right? So these air cavities, why it is called air cavities? Because it is filled with the air. It is filled with the air, and air is having both the carbon dioxide as well as the oxygen. And these are the two things that are required in the process of photosynthesis and respiration, respectively. Now, vascular system found in the veins and the midrib. These vascular bundles are found in the veins and midrib region. The vascular bundle size depend upon the vein size. What is the size of the vein? This vascular bundle depends on that. Let us see the different features of the vascular bundle. Surrounded by the thick-walled bundle sheath cells, and xylem position can be identified with the within the vascular bundle. Now, if you see the venation, type of venation in the dicot leaf, then it is having the reticulate venation, right? So this is the midrib region. Okay, you can see the xylem region, you can see the phloem region, right? So this is the midrib region. And when you see from the midrib, veins are given out. So veins will have this type of vascular bundle, but the size of the vascular bundle will be small. Okay, the vascular bundle size will be small in the veins as compared to the midrib. And there will be the veinlets also. If you see the veinlets vascular bundle, then it will be again very small. Okay. Now we will talk about the monocotyledonous leaf. Similarities of the dorsiventral leaf. Resemblance dorsiventral leaf in many ways. What are the characteristic differences? Stomata present on both the surface of the epidermis. See, in the case of dicot, the stomata are confined to the lower surface, but in the case of monocot, they are present on both the surfaces equally. Mesophyll cells not differentiated into palisade and spongy parenchyma. This is a very important feature of the monocotyledonous leaf or the isobilateral leaf. Why it is called isobilateral leaf? See the grass, their leaves are not dorsiventral because you cannot say this is the dorsal surface and this is the ventral surface. They are mostly erect. They are mostly erect. And that is why they are called as isobilateral, right? Not differentiated into palisade and spongy parenchyma. So here you can see. You can see the epidermis, right? Here you can see the epidermis. You can see the stomata on the upper surface. You can see the stomata on the lower surface. This is very important feature. Now, see the mesophyll cells. No palisade parenchyma. All the circular cells are there, right? And the air cavities are less as compared to the dicot leaf. So these are the mesophyll cells. See the xylem arrangement. The xylem vascular bundles are quite larger in size and it is present from the epidermis to the upper epidermis to the lower epidermis, right? See this. And there is a like this ridge also on the upper surface where the midrib is present. This vascular bundle showing the midrib region actually. You can see the xylem. You can see the phloem. This is the phloem part, right? Now, there are some special features in grasses. Bulliform cells. Bulliform cells, modified adaxial epidermis cells along the veins. These are some of the cells which are present on the adaxial epidermis cells along with the veins. They are very, very important cells. They are large, empty and colorless. Okay. When they hold, means when these bulliforms fold, okay, they minimize the transpiration. What is the function? Turgid bulliform cells exposes leaf surface. When this bulliform cells are filled with water in a turgid condition, they expose the leaf. They expose the leaf so that they could get the maximum sunlight and do the process of photosynthesis properly. But when this bulliform cells in a flaccid condition, it means when the plants want to conserve the water. Water availability is less in the soil. So at that time, what they do, these flaccid bulliform cells closes or curl the leaves inner side, right? So they are helpful in the conservation of water. Okay, where they are present, this is very, very important. Adaxial epidermis cells along the veins. Remember this line. Parallel venation. What type of venation we see in the case of monocot? Parallel venation. Vascular bundles are of similar size except the main vein. Okay, the main midrib is the main vein and they are having yes, it is the same. Water loss is due to the exosmosis that get the leaf flaccid. Okay, in that way only. Osmosis is there. Okay, the vascular bundles of similar size except the main vein. Observe in the vertical leaf section. Here you can see this is the main vein. And when you see the parallel veins, this is the midrib region. When you will see the parallel, they will be quite smaller compared to this midrib. That vein will be having smaller vascular bundle. Now stem summary. Dicot vascular bundles is a ring and open type and they are having the cambium present. Cambium is present. Monocot, scattered vascular bundle and they are not having any cambium. So they are closed type. Leaf, if we talk about the leaf, dicot and they are having the dorsiventral leaf. Different upper lower surfaces. Distinct palisade and the spongy tissue they are having. What about the monocot? Isobilateral. This leaf is called isobilateral. Similar surfaces and uniform mesophyll tissue. Only one type of mesophyll tissue is there and they are having the parallel venation. Now there will be some NCERT based question. Why is the monocot stem unable to undergo secondary growth? Let us see the answer. Monocot stem cannot undergo secondary growth due to absence of vascular cambium. They are not having any vascular cambium. So the vascular cambium actually divide and redivide the cell. But here, if this cambium layer is absent, this meristematic tissue is absent, so there will be no secondary growth. Monocot lack vascular cambium, which is essential for secondary growth. Without cambium, no new secondary growth or the phloem can be produced, because cambium divide towards the inner side and outer side, and they will form the xylem and phloem respectively. Scattered vascular bundles. Vascular bundles are scattered throughout the stem irregularly. So no organized arrangement in the rings preventing the coordinated secondary growth. This thing I was discussing when we were discussing about the difference between the cortex region of the monocot and dicot, right? And actually they do not require this secondary growth. Okay. Closed vascular bundle. The vascular bundle are closed type. It means meristematic tissue between the xylem and phloem. Fixed bundle structure. Once formed vascular bundle cannot expand or multiply. They will not multiply here because vascular bundle is fixed. No provision of adding new conducting tissue, because new conducting tissue means new xylem and phloem. That will not form here. Totally absent. Growth limitation is there. In the case of monocot, primary growth is only means the development of the stem. Increase in girth occur only in the cell enlargement, not the cell division. The cell can enlarge. The cell can enlarge. The cell can expand. And that can bring about a little growth in the width of or the girth of the plant or the stem. But it will not be similar to the one which we see in the case of dicot plant due to the secondary growth, due to the division of the cells. Okay. And every year, this is also one of the things that most of the dicot are perennial plants. And what about the monocot? They're not perennial, right? Next question. What are the main differences between the anatomy of dorsiventral and isobilateral leaves? Let us see the answer. So there are so many differences. Let us see. Mesophyll. Regarding mesophyll, differentiated into palisade and spongy tissue. Both the surface have palisade tissue, spongy tissue in the middle. Palisade layer. Present only upper adaxial surface. Present on both the upper and lower surfaces. Stomata. Only on the lower surface. And they are present on both the surfaces. Chloroplast distribution. More in palisade cells. Equally distributed in the cells. Leaf orientation. Horizontal into the ground. Means dorsiventral. Vertical to the ground. Typical example. Most dicot leaves, and here eucalyptus and many monocot. Upper and lower surface. Structurally different. If you see the upper surface and lower surface of the dicot leaf, they will be different. But in the case of isobilateral leaf, monocot leaf, their surface will be similar, quite similar. Light reception, mainly from the upper surface. And here, as they receive the light from both the surfaces, right? Because they are in vertical direction. So they receive the sunlight from both the surfaces, right? Keynotes. The vascular bundles from the ring pattern that allow the secondary growth through the cambium activity. Contain distinct pith and cortex region in the dicot stem. Unique feature, presence of interfascicular cambium between the vascular bundle. So when there is a secondary growth, some of the parenchymatous cells that is present between the vascular bundle, they behaves like a cambium and they will form a full grown cambium layer. As a stellate star-shaped pith in some species, possesses active cork cambium, monocot stem, scattered vascular bundle with no specific pattern, and unique characteristic, presence of ectostele, scattered stele, means scattered vascular bundle. Each vascular bundle is surrounded by the sclerenchymatous bundle sheath. This is very important for the protection of the bundle sheath. No distinct pith and cortex separation here, and generally lacks secondary growth due to the absence of cambium. Dorsiventral leaf, distinct differentiation between the upper and lower surface. Unique feature, distribution of stomata mainly on the lower surface. Hypostomatic, this is this condition. Contain distinctly elongated palisade and parenchyma cells. Reticulate venation pattern we see. Presence of bundle sheath extension in some species. Now regarding the monocot, similar structure in both upper and lower surface. Parenchyma is of similar type. Stomata present on both the surface, that is called amphistomatic. Parallel venation with regular intervals, and presence of bulliform cells in help in leaf rolling. Specialized crans anatomy in the C4 plants. Some interesting questions for you. Compare the epidermal features of the dicot and monocot leaves. Draw a label diagram of the TS of A, dicot stem, and B, monocot stem. Explain the significance of A, bundle cap, bulliform cells, pericycle, and how does the position of xylem and phloem differ in the dicot and monocot stem. So these are some important questions that you can solve with your. So this is all about today's session. Thank you very, very much for joining today.